Hello everyone, my name is Valerio Laghi and I work as a researcher assistant in Institute Pasteur. In this unit, I will give you an overview of modeling an infectious disease in zebrafish. Studying the interaction between host and pathogen is never easy, especially if we are talking about viruses. To understand the propagation of a virus inside patients, we almost always need to infer indirectly the path and speed of the propagation from blood to samples. For example, if we are trying to reconstruct the virus propagation in a human body, we need to take samples at different time points, which is feasible with blood, but almost impossible with other organs and tissues. From these blood samples, we can observe the virus concentration in the tissue X, for example, at time t, and using our biological knowledge, we can try to build our hypothesis about the virus behavior. This kind of study takes a long time, gives just an outlook of the viral propagation and need to be repeated many times to have a solid statistic baseline. If we take in consideration the complexity and heterogeneity of all the different components that interact with the pathogen during infection, having only relative concentration of viral particles and other components is not enough to have a solid model in a short time. The mathematical modeling of infection allows us to deduce from this scarce data far more. We can use the mathematical modeling to insert in a wide frame every data that we have, and like a puzzle, fit every piece, finding what we are missing or deducing the missing pieces. For example, in 1995, came out an article breaking the misconception that HIV, similarly to other retrovirus, enter a latency phase. In this article, through modeling, was demonstrated that HIV infection is really a dynamic process. It was demonstrated that the HIV was rapidly and constantly replicating and getting cleared from the immune system. This turnover with the normal investigation method explained before will give an average virus concentration constant in time, bringing the researcher to think that the virus was dormant. Understanding this rapidly error-prone replication gave the impulse to change therapies, circumnavigating the predicted drug resistance and speeding up the usage of drug cocktail. The effect of modeling is tangible and relevant. In 2015, a paper about ACV demonstrated that it is possible to model the effect in vivo of drugs in clinical trial in just days, a lot less if compared to the precedent average of 48 weeks. This led to the faster developing of new therapies, and in these therapies we can find a successful interferon-free DAA. Even though we will not discuss in detail the big data modeling in this unit, it's worth to mention it. The database of clinical and biological data about virus propagation and dynamics are increasing, and the mathematical model can give a structure to said data. Besides tying together data with different nature, mathematical modeling on big data can mine out models that can adapt to different viruses, show new relevant correlation, and identify drugs that can work on another virus besides the one they were developed for. The potentiality of big data modeling is high and will be a powerful tool for virologists in the future. Another interesting property of the mathematical modeling of host-virus interaction is the possibility to determine basic behavior patterns that can be used horizontally by other viruses. A basic explanation is that if the model fits, this means that the virus behaves in a ready to this way, and we can speed up the work on it. But if it doesn't fit, we can identify why and focus the work on the new founded characteristic of the virus. About the host, in the last couple of years, the zebrafish is entering the spotlight. The reasons are related to its ability to bridge in vitro and in vivo experimentation, bridging high-throughput studies and whole organism studies. Furthermore, the innate immune system of zebrafish is really similar to mammalian one, presenting an interferon-based innate immune system with antiviral functions. If we couple this with overall protein and tissue similarities, we can use this animal model to follow the viral propagation over time and create a solid statistical basis for mathematical modeling, speeding up the process of realization of the model. Zebrafish is easily manipulable, so we can modify parameters in the host-pathogen interaction and then challenge the mathematical model to further ameliorate it. As we said before, the biggest problem in following the viral propagation is to observe directly the virus movement. Zebrafish offer high optical transparency that can be used to observe directly the virus propagation over time. Coupling the interferon protein with a fluorescent report and labeling the virus, we can do in vivo experimentation and observe the timing, intensity, and directionality of the host-virus interaction and virus propagation. This makes the zebrafish a powerful tool to increase the resolution and production speed of mathematical models. This is an example of basic model of viral replication. In this model, established by Jeremy Gesh, we can observe the presence of E, infected cells, and T, target cells. Using this model, we can infer several different parameters, like the ratio of rising of new infected cells, the production of new virons, and the ratio of cell death and clearance of virus. Normally, parameters like the number of virions produced and lost, for example, are not easily determinable by experimental procedures, but with this model, it's still possible to do it. From a seemingly simple model, it's possible to build much more complicated models that take into account variables like the interferon response or the crossing of the blood-brain barrier. We can start from the core model and solidify it, and later add layers of complexity based on our first batch of results. This shows that the models are highly adaptable and can answer directly or indirectly specific questions. It's important to understand that often the model itself is used to describe and investigate what happens inside a biological black box, so it can be used not to investigate results, but to investigate what brings to said results. Modeling brings together and involves different fields in the effort of describing what we already know about infection mechanism and uncover what we cannot see directly with experimental procedure. Viral infection on zebrafish can reduce the time necessary to pry open the black box immensely, reducing and focusing the burden of mice and human experimentation, only using the model for the final fitting. I hope you enjoyed the last session of our MOOC. See you around.